Good morning. At the beginning of the semester, we discussed the fact that what this course was really all about was looking at mechanisms of gene expression and how genes function within the cell and within an organism. We talked briefly about the flow of genetic information in that DNA was considered the storage molecule for that genetic information or the instructions in order for a cell to survive and function. We talked about the flow of that information going from DNA to RNA and RNA to protein, and then some final functional phenotype that could be assessed. This is the classic flow of information referred to as the central dogma. We also at that point introduced a few terms, a gene, gene expression, and the different levels of gene expression, including transcription and translation. What we did in the last two lectures in this unit of material was talk about the structure of DNA from the nucleotide level all the way to the organization of the DNA within the cell in question. What we are going to transition into with this particular lecture is looking at the mechanism of replication. How is it that all of the genetic material is copied within a cell in order to prepare for either mitosis or meiosis? And that is the process, again, referred to as replication and the focus of today's lecture. Typically, when I discuss the process of replication, that is the copying mechanism of the genetic material, again, as the cell prepares for either mitosis or meiosis, I like to go back and talk a little bit about the work of Watson and Crick. Watson and Crick's structure presented more questions to the scientific community. The first thing I want to review is the concept of complementarity, that is the chemical affinity provided by hydrogen bonding between bases. We know that there are specific hydrogen bonds or hydrogen bonding that occurs between specific bases, that is, A's, nucleotides that contain adenine, or A's, will always hydrogen bond with nucleotides that contain thymine, or T, and that there are two hydrogen bonds between A's and T's, and nucleotides that contain guanine will pair up or hydrogen bond with nucleotides containing cytosine with 3 hydrogen bonds. This is again referred to as sequence complementarity. We also know from previous work that we did not yet discuss that the two strands were actually anti-parallel. Anti-parallel just means that the two strands in the complex double helix shape are oriented in opposite direction of one another. Now, one of the things I like to mention also is that Watson and Crick published a paper almost immediately after the paper that described the structure of the DNA molecule. In this paper, it contained absolutely no data, but only speculation. One specific quote from that second paper that was published is listed on this slide. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism. What they proposed in this paper was that because of the sequence complementarity, if you could simply pull the two strands apart from one another, because of that specific sequence complementarity, if you know the sequence of one strand, you immediately can deduce what the sequence of the second strand is. And this is what they proposed happens during DNA replication, that somehow the strands separate and that each strand acts as a template for the missing information. Now, they didn't have any research to back up this particular speculation. They just proposed that this was most likely how DNA replication was occurring. So at this particular point in time, there was different models or modes of replication that were proposed by different groups of scientists. One model for how DNA replication occurs is referred to as the conservative model. And in the conservative model, yes, the two strands do have to separate, but that somehow the newly synthesized DNA and the old original DNA remained together. So if you look at the slide with the figure, the conservative model shows the original DNA strand in pink, and then after one round of replication, you have two double-stranded DNA molecules, but the pink representing the old or original DNA strands are still together, and the blue representing the new synthesized DNA is also together. So in the conservative model, this is what's being proposed. Another model at the time, which is what Watson and Crick had proposed, is referred to as the semi-conservative mode or mechanism of replication. With the semi-conservative mode or model of replication, the two strands separate, again represented by the pink, and at the end of a round of replication, you again have two double-stranded DNA molecules, but this time each of those double-stranded DNA molecules contains one old or pink strand of DNA and one new or blue strand of DNA. Lastly, there were some scientists that proposed a dispersive model, and in a dispersive model, what you would have is the two strands that result at the end of the round of replication are some combination or mixture of new and old. So one strand is not entirely new, one strand is not entirely old, like what's proposed by the semi-conservative model, but that each of the strands in each of the double-stranded molecules are some mixture of old and new. In 1958, it was the work of two scientists, Michelson and Stahl, and they conducted a series of experiments that then demonstrated that the semi-conservative model or mode of replication was in fact the way DNA replication was taking place. We're not going to go into those complex experiments, but I did want to give Michelson and Stahl credit because their experimentation and their research was able to demonstrate that yes, in fact, what Watson and Crick had proposed as the semi-conservative model was in fact the way DNA replication was occurring. The next two slides are representations or overviews of how DNA replication is in fact taking place. So focusing on this particular slide, on the left-hand portion of the figure, you have the single strand of DNA in dark blue, which is labeled the template strand. This is the strand that is being utilized as the information to synthesize the new strand. The new strand being synthesized all the way on the far left in the lighter color blue. Based on that semi-conservative mode or model of replication, the two original strands have to separate and each strand acts as a template. So here you have again the dark blue representing the template strand and the newly synthesized strand in the replication process in the light blue. What is important to note that what is required for DNA synthesis is referred to as a nucleoside triphosphate. This is the building block required to synthesize DNA. It is, in generic terms, a nucleotide, but more specifically, a nucleoside triphosphate. Again, sugar, base, and 3 phosphate groups is required as the building block. What happens is the base pairing occurs between the correct incoming nucleoside triphosphate, in this case one that contains T or thymine, and the nucleotide that is in the template strand, in this case the one containing the A. So based on that sequence complementarity, this is what dictates what nucleotide comes in. The addition is going to be the 5' end of the incoming nucleotide is going to attach to the 3' end of the growing chain. Then on the far right, you see that nucleotide or nucleoside triphosphate has been added in. In the process, one phosphate from that triphosphate molecule remains as part of the structure of the new strand being synthesized, and a diphosphate, known more specifically as pyrophosphate, is released in the process. It is this release of the pyrophosphate that provides the energy that drives the reaction of synthesis. Any type of synthesis required in the cell typically requires an input of energy, and in this case the energy comes from the release of the pyrophosphate from the triphosphate nucleotide or nucleoside triphosphate. Again, that energy is what drives the reaction of synthesis. So if you provide a cell with monophosphate form of a nucleoside or a diphosphate form of a nucleoside, DNA synthesis will not occur. It must always be the nucleoside triphosphate to drive synthesis. And this figure is just another view of that same process, where you have the template strand sort of highlighted with the grayish color, and then the newly synthesized strand highlighted in the lighter green color. The important thing here, exactly like what we saw on the last slide, is that you have a nucleoside triphosphate that is the requirement to synthesize DNA. It's the building block that's required, and that the 5' end of the incoming nucleotide is going to be covalently linked to the 3' end of the growing chain. This 5' end and 3' end designation we went over in our previous lectures, and it's really important to understand that as this concept of 5' to 3' synthesis, or 3' to 5' directionality, is going to become really important. So knowing the 5' end versus the 3' end, as I emphasized in that last lecture, is extremely important. Now, based off of what we've just discussed, it's been established that DNA replication occurs in a semi-conservative fashion. But once that was established, there were additional questions that quickly came about as far as replication is concerned. One question that was asked and eventually answered was, does DNA replication occur at random locations, meaning the initiation of replication occur at random locations, or does it occur at a specific site? Mutational analysis, that is where DNA sequences are altered, were utilized to answer this question, and that is that DNA replication occurs at a specific location. That is, the initiation or the start of replication always occurs at a specific location. In prokaryotes, and the most common prokaryotic example is Escherichia coli or E. coli, that replication origin is referred to as ORIC, and we talked a little bit about replication origins in our first unit of material, specifically dealing with plasmids and allowing plasmids to be copied or replicated overall. They do in fact need a replication origin. So again, the answer to the question is there is a specific set of sequences that are required for the initiation of replication. These are referred to as the replication origin, and in E. coli it is specifically referred to as ORIC. In addition, another question that came about was, is replication occurring in one direction or in two directions away from that replication origin? Another way to put that is replication unidirectional or bidirectional. So going back to prokaryotic chromosome structure, we discussed that prokaryotic chromosomes are found in a circularized structure. So imagine if you have a circle and you have a starting point of replication. If replication occurs in one direction, let's say clockwise from that position, you would imagine that replication would continue all the way until it came back to the same starting point. However, bidirectional replication would be replication that started at the replication origin but moved away from that replication origin in both directions away from the starting point. In that model, what you would expect then is those two ends to meet up at the opposite location of the circle. And this too was done by a number of different types of studies, including the use of radioactive material to determine where active replication is taking place. And if you look in particular at the top figure in this particular slide, what you're looking at is the use of radioactivity to, again, detect where active replication is occurring. So at the very top of that figure, you see the replication origin. And as you continue down that figure over time, what you're seeing is that replication is actually moving in opposite directions away from that replication origin, and active replication is at both of those locations. Additionally, I'll add some definitions here. The replication fork is the point along the chromosome where active replication is taking place. So in the same figure we were just talking about, it's that orangish reddish color where active replication is occurring is referred to as the replication fork. So in this figure, there are two replication forks, again, moving away from that origin where replication was initiated. The term replicon refers to any DNA that's under the control of a replication origin. For most prokaryotes, the entire chromosome is the replicon because that's the DNA that's under the control of that single replication origin. We're going to eventually talk about eukaryotes, and what we'll hear about when we discuss eukaryotes is that there are multiple replication origins. So any DNA that falls under the control of a single replication origin is considered a replicon. So many eukaryotes have multiple replicons because they have multiple replication origins. Oftentimes what I like to do is give an overview of a process and then go back and piece it apart. This particular figure you'll come back to, or I hope you come back to and review when we start to look at all the small components associated with replication. What I do want to highlight is that replication is divided into three broad stages. On this slide, you can see initiation, which is how the process gets started, elongation, which is the continual addition of nucleotide after nucleotide after nucleotide. And then on the next slide, you'll see the third phase, which is termination. How does the process all stop? What you can see when you focus on this particular slide, at the very top figure is highlighted in pink or red, is the ORI, or the replication origin. So, specific set of sequences that are utilized to initiate replication. What happens is there are a whole series of enzymes that will associate with the replication origin to initiate the process of replication. So on this slide, don't worry necessarily about the names of the enzymes because we're going to look at them in more detail, but I do want you to come back to them again as we start to talk about them, or after we finish talking about them. All right, so enzymes, we're going to keep it broad right now, recognize the replication origin to open up the double-stranded DNA and begin the process of replication. Elongation, like I mentioned just a minute ago, is the continual addition of nucleotide after nucleotide after nucleotide. And then on this slide, we transition between elongation to termination. Termination is where the process is going to stop. What you can see highlighted on this slide is that there is actually a set of sequences that are the termination site, and we'll come back to that in just a little bit. So again, this was just meant to be a quick overview. what we're going to do is we're going to piece apart all the individual stages and the enzymes involved in those stages. What you should do in part of your studying mechanism is go back and review these two slides after we've looked at individual pieces and parts and put them all back together again. So as mentioned in previous lectures, what I typically like to do with most of the mechanisms that I focus on is to highlight what is going on in the prokaryotic model, which is usually E. coli, and then add in some eukaryotic examples and some expansion with what's going on in eukaryotes. But I really want you to focus on the mechanism and understanding the mechanism in prokaryotes, because in general they are simpler than what's happening in a typical eukaryote. So for DNA replication in E. coli, there are over 30 different enzymes, polypeptides that cooperate to get DNA synthesis or DNA replication to take place. So right there you can see that it's actually not very a simple process if you've got over 30 different additional components helping the process along. Luckily for us, we're not going to learn all 30, but we are going to highlight some very important players in the replication process. So we're going to start off with an enzyme known as DNA polymerase. Anytime you see the ASE ending on a word, that's going to indicate that it's some type of enzyme. So DNA polymerase is an enzyme that makes DNA polymers. Makes sense. In the late 1950s, 1957 to be exact, we have the work of Arthur Kornberg and his associates. What they were able to do is isolate an enzyme from E. coli that could direct DNA synthesis in vitro. And in vitro means in the test tube. With this enzyme, they tried a series of experiments and found that there were some requirements for DNA replication to occur with the enzyme that they isolated. I should add that they named this enzyme DNA polymerase, but more specifically DNA polymerase I. What they found from their series of experiments was that the building block necessary to synthesize DNA, like I mentioned in the previous slides, is a nucleoside triphosphate. Diphosphate nucleosides and monophosphate nucleosides do not work. You have to have all 4 nucleoside triphosphates in your reaction in order to synthesize DNA. This makes sense. If you're going to make more DNA, you need the building blocks to make that DNA. And in this case, the building block does need to be the triphosphate nucleoside. In addition, from these experiments, they also found that you did in fact need to have some sort of template DNA. That this enzyme did not just synthesize DNA de novo, meaning from the start. It doesn't just take random nucleotides and link them together to make DNA. There has to be some template molecule in your reaction. And of course, you do need the enzyme in order to synthesize the DNA. In addition, what they found was that there were some naysayers or some that doubted their work and said that this enzyme that they isolated synthesized DNA in the test tube, but does that really mean it's making a biologically active molecule? They did in fact complete some additional experiments where they synthesized a bacteriophage genome, and bacteriophage is a virus that infects bacteria. They synthesized it in the test tube, they isolated it, and they utilized it to infect E. coli. What they found was that this DNA that they synthesized in the test tube initially was able to produce new phage. And so that suggested biological activity to the DNA that was synthesized in vitro or in the test tube. Okay, let's look at DNA polymerase in a little more detail. What you can see on this particular figure are two different views of the DNA polymerase molecule. On the left-hand side, you're looking basically at the ribbon model for the enzyme. And then on the right-hand side in panel B, you see more of a cartoonized version of the enzyme. Regardless of which version you're looking at, I tend to like to look at the cartoon version because it's a little more simplified overall, is that DNA polymerase has some very distinct structural characteristic features that have been established basically from X-ray crystallography and other types of microscopy tools to establish the structure. So when we talk about DNA polymerase, there are some components. There are what's referred to as the fingers and the thumb. The idea here is that if you had your hand sort of cupped in a C-like shape, this represents the structural characteristics of DNA polymerase with your thumb and your fingers cupped overall. And so if you can imagine that with this and superimpose your hand over the structure, this is what the molecule looks like. In your hand, you have the palm portion, and this is referred to as the catalytic site, or what I oftentimes call it the place of business. This is where the addition of nucleotides is taking place. There is a requirement for this enzyme of a divalent cation, which is usually magnesium or zinc. These are allowing the enzyme to function overall, and if you reach back into your basic cell biology, are oftentimes referred to as cofactors. Many enzymes require cofactors in order to function, and DNA polymerase is no exception. The fingers portion of the enzyme is a portion that binds incoming nucleoside triphosphates and closes in to bring those nucleoside triphosphates in. So the structure is not static, it's actually dynamic and constantly moving. So again, if you use your hand sort of in a C shape and then, or like a Pac-Man, and start closing and opening, that's actually what's happening during the process of replication, is that the enzyme is moving as the synthesis is taking place. There's also the thumb portion, and the thumb portion helps to interact with newly synthesized DNA, usually stabilizing it, sort of helping maintain the correct position, correct interaction between DNA and polymerase overall. What I do like to mention at this point is that this particular enzyme is actually extremely fast. It typically adds 1,000 nucleotides per second. And so that's actually a very fast-acting enzyme overall, which you would imagine needs to be fast in order to replicate the entire genome of a given organism. Now, one of the things that I mentioned on the previous slide, or one of the previous slides, was that DNA synthesis always occurs in the 5' to 3' direction. That is, the 5' end of the incoming nucleotide attaches to the 3' end of the growing chain. So DNA synthesis, again, always occurs in the 5' to 3' direction. It turns out that this enzyme that they isolated actually has some other functions associated with it. There is exonuclease activity with DNA polymerase 1. So let's look at the word exonuclease and piece it apart just a little bit. If we look at the root nuclease, again, it has the ASE ending, which tells us it's some type of enzyme. And a nuclease is a broad class of enzymes that degrade or break down DNA, typically one nucleotide at a time. We talked about nucleases, in particular restriction endonucleases, when we had our first unit of material, so this shouldn't be completely new to you or a new concept to you. We also highlighted at that point the difference between a nuclease that is an exonuclease versus an endonuclease. Now, those restriction enzymes are endonucleases. They recognize a specific sequence and make a cut. What I want to highlight here is something referred to as an exonuclease. And an exonuclease is a nuclease, but it's going to be one that cuts on the end of the molecules and not internally. So it's working its way from the end in one direction or another. It turns out that DNA polymerase has 3' to 5' exonuclease activity. The 3' to 5' exonuclease activity is also referred to as a proofreading function. This is a nuclease activity that allows for the removal of a nucleotide that was inserted incorrectly. So the whole premise behind DNA replication is that sequence complementarity that we keep coming back to. What dictates what nucleotide is going to get added into the growing chain is what nucleotide, specifically what base, is found on a nucleotide in the template strand. So in this top figure, you can see the black indicates the template strand and the blue indicates the newly synthesized DNA. In the template strand, we have a G that should have the next nucleotide added to. And what you can see is that in this example, an A-containing nucleotide was added in. That's an incorrect base pairing because G's should always hydrogen bond with C's. That can be recognized by the enzyme as a distortion. There's no base pairing that's occurring, so the wrong nucleotide got added in. And what it can do is cut that nucleotide out. So 3' to 5' exonuclease activity, cutting that nucleotide out. And now can continue with the DNA synthesis process. In addition, this enzyme can also have 5' to 3' exonuclease activity. This would be an exonuclease activity that works in the 5' to 3' direction. Usually that activity is involved with DNA repair, and we will come back to that later in today's lecture. Not necessarily repair, but what the function of this portion of the enzyme is. This particular slide just highlights that 3' to 5' exonuclease activity that I was discussing in the previous figure or previous slide. Again, just another view of that to understand that the incorrect nucleotide that has been added in can be cut out or removed, and then DNA synthesis can continue. All right, so now let's go ahead and jump into getting an understanding of how this replication process actually occurs and talk about some of the key players or enzymes that are involved in the process. We know that in E. coli, there is a single replication origin, and it is referred to as ORI-C. ORI-C is a total of 245 base pairs, and it is made up of repeating blocks referred to as 9-mers and 13-mers. So in this figure on the far left, you can see the color-coded blocks. A 13-mer is just a short hand or slang way of saying a block of 13 nucleotides. And a 9-mer is just a short or slang way of saying a block of 9 nucleotides. What you can see here, though, is that those 13-mers are repeated, so there's that same sequence, the same red color repeated multiple times, and the 9-mers, the same sequence repeated multiple times. And this represents the replication origin. With the replication origin, this is again the start or the location where replication begins. In this slide, I'm going to overview things, and then in the next several slides, there are some reviews of the same topic, just different figures overall. So what I want to start off with then is, after we've talked about the structure of the replication origin, the first enzyme to come into play is an enzyme known as DNAA. DNAA in this particular figure is those cream-colored circles, And these are proteins that bind to the 9-mer sequence. They are proteins that are DNA binding proteins, so they can recognize the specific sequence and bind to the DNA. But they also have protein-protein binding interaction, meaning that they can bind to each other. So you see DNA-A, multiple copies of DNA-A coming in and binding to the 9-mer sequence, but hopefully what you can see is they're also binding to each other. This begins to form what's known as a tension bubble, in that it causes a bend in the DNA and causes tension in the DNA. The 9-mer sequence happens to be rich in A's and T's, which only has 2 hydrogen bonds between each A and T. So when the DNA is bent because of the binding of DNA-A, that then causes tension and forces the popping open of those 13-mer sequences because they are rich in A's and T's. You now have what's referred to as an open complex. You then have a pair of enzymes coming together, DNAB and DNAC. DNAB is an enzyme known as a helicase. Its job is to unwind DNA, and DNAC is the helicase loader, or helps regulate the helicase. So they do work together, but the actual helicase is DNAB. Again, what happens is this complex jumps on to the replication bubble or opening and starts to unwind more of the double helix. You also have an enzyme known as topoisomerase, also listed in this slide as DNA gyrase. This is an enzyme that makes a single cut, and again, we've talked about topoisomerases previously when we talked about DNA structure. It makes a single cut, which allows a swivel point, because as you begin to open up the replication bubble, you create tension on either side of that opening, and tension could lead to damage of the DNA, which would not be good. So by adding these single cuts on either side of the opening, it allows a swivel point and allows for the release of that tension. Lastly, what's not shown in this figure but is listed on the slide is single-stranded binding proteins, and those single-stranded binding proteins bind to single-stranded DNA and prevent the closure of the single-stranded DNA. So the next 2 slides are just the same thing we talked about with the addition of enzymes, just another view. And I always like to add different views because conceptually I think it helps to understand different interpretations of the way this is happening. So in order for initiation to occur, the strands do in fact have to separate. How do those strands separate? With the very first enzyme coming into play, known as DNA-A, which has the ability to recognize those 9-mer sequences, in this particular slide listed as DNA boxes. Those proteins, DNA-A, represented in the green, binds to the DNA boxes, or the 9-mer sequence, and begins to cause tension. That tension then causes the popping open of those AT-rich elements, or 13-mer sequences. Then, like on the previous figure, you have DNAB and DNAC come in together to play a role in the next step. DNAB is the helicase, which is an enzyme that unwinds DNA, and DNAC is an accessory to help that enzyme along. So the helicase unwinds the DNA and makes the replication bubble even larger. What I like on this particular figure, down at the bottom of the figure, is you can see 2 helicases are added to either end of the opening. Remember, we've already discussed that replication occurs bidirectionally, moving in opposite directions from the replication origin. So you're going to need 2 sets of enzymes on either end of that opening to continue the process, and that's really nicely shown in this figure. So at this point in the replication process, we have a replication bubble that has formed. which means the two strands have been opened up, and each one can now act as the template for the synthesis of the missing strand. Interestingly, DNA polymerase cannot just recognize a single-stranded template. It actually has to have an attachment point to attach nucleotides to. So even though we've now separated the two strands, those two strands cannot initiate DNA synthesis without the help of an additional component, which is what we're going to focus on for this slide. The enzyme involved in adding that additional component is known as primase, also known as DNAG. Primase is a type of enzyme known as an RNA polymerase. It is an enzyme that synthesizes RNA. In this case, primase synthesizes a short stretch of DNA known as a primer. A short stretch of RNA known as a primer. That's represented in the top figure in the orange. So primase synthesizes a short complementary sequence of RNA, but this now provides an attachment point for the DNA polymerase to attach DNA nucleotides. So after the short primer is synthesized, DNA polymerase can now recognize this primer template combination and start adding nucleotides one right after the other. An additional enzyme that we have not yet mentioned is an enzyme known as the beta clamp or the sliding clamp. This particular molecule has a donut-like shape to it, and it is a molecule that binds to DNA polymerase and allows for the DNA polymerase to stay on track with association of the template. So again, this keeps the enzyme in position and allows for efficient DNA synthesis to occur. Otherwise, the polymerase oftentimes, if it's not held in position, would synthesize a few nucleotides and simply fall off. So this addition of the clamp binds to the polymerase and keeps them on track, on target for DNA synthesis. Now, because the two strands in the double helix are anti-parallel, this presents a problem with replication. It's a conceptual problem. If you look at the figure at the top, in the right-hand panel, you can see basically what's a time lapse, if you want to call it that. So earlier in replication, a little more along replication, and then further along in the replication process. If all of the enzymes are located right at the fork, which is sort of the Y-like structure where there's a difference in directionality, what you can see is that one strand, once it's initiated, this is the bottom one, will just continue. As long as the strands are unwound, DNA synthesis will continue. So looking at just the bottom strand in each one of those progression of figures, as long as the template is opened up, DNA synthesis can occur continually. This is referred to as continuous DNA synthesis and occurs on what's known as the leading strand. However, because the strands are anti-parallel, the top strand in this double-stranded DNA molecule is going in the opposite direction, and hence DNA synthesis has to move away from the replication fork, like you see with the point of the arrows. What happens in synthesis here is short bursts of discontinuous synthesis, each one requiring their own primer for the synthesis to initiate. So this occurs on what's known as the lagging strand and is referred to as discontinuous synthesis. This was all experimentally demonstrated in the late 1960s by Reggie Okazaki and his colleagues. They utilized a series of experiments that also incorporated radioactivity and basically found that there was, in fact, some synthesis that occurred continually and some synthesis that occurred in a discontinuous fashion. Those small fragments that are synthesized in the discontinuous fashion are referred to as Okazaki fragments. All right, now let's sort of put all of those pieces of the puzzle together for the elongation process. In this first figure, what you're seeing is how complex this mechanism actually is. So in the figure, in the gray, on the right-hand side is the original double-stranded DNA. And then on the left-hand side, you can see the 2 strands of double-stranded DNA that are being produced during active replication. What are the enzymes that are involved in this process? Well, you have DNA polymerase in the light green, and you can see that for each of the strands, there is a separate DNA polymerase. There is the single-stranded binding proteins, which we didn't talk too much about on the last set of slides, but you can see those single-stranded binding proteins binding to prevent closure of the single-stranded DNA back to the double-stranded DNA form. Each of the polymerases has their own sliding clamp, which's job is to keep the polymerase on track. You can see in the orange in the center the DNA helicase, which is going to unwind the DNA. And then lastly, the larger structure right in the center is known as the clamp loader. Its job is to attach the sliding clamp to the polymerase while intact, or keeping it intact with the DNA. With this particular figure, what you're seeing is the primase coming in and synthesizing the RNA primer, which is going to be required for the Okazaki fragments on the lagging strand, which again will produce discontinuous DNA synthesis. You can then see the primase is released. The primer has been synthesized in the greenish color, and there's going to be a new association of polymerase with clamp at the site of the primer, which will be on the next figure here. So here in this next figure, you can see the clamp loader loading the clamp at the site of the primer, and what it's going to do is actually bend and allow for its association with the DNA polymerase. And then in this figure, you see that interaction with the polymerase and the primer, ready to go ahead and synthesize the next stretch of DNA. Now, lastly in this process, we want to talk about termination. There is a specific location where termination occurs, and I'm not going to get into that into too much detail. What I do want to talk about, though, is that there's a problem with DNA replication in that in particular with the discontinuous synthesis, every time a short burst of DNA is synthesized, there is a requirement for a primer. That primer is an RNA-based molecule, so throughout the newly synthesized DNA, there are locations where there are DNA-RNA hybrids, and that RNA needs to be removed and filled in with DNA. There are a series of enzymes that play a role in helping this process along. In this particular slide, you see the enzyme known as RNase H, which recognizes DNA-RNA hybrids and removes the RNA. DNA polymerase also has that exonuclease function that we talked about, the 5' to 3', and so it usually cleans up that space to where you now have an open block right there in the center of the slide. And then DNA polymerase can fill in that missing information with DNA. And then finally, the last enzyme to play a role in this is DNA ligase. It closes up that covalent linkage to make a smooth, seamless piece of DNA.